Hello everyone, we're going to start the meeting now. So a very warm welcome to the 18th annual Complex Trait and Rat Genomics Conference, live here from Manchester, UK. I'm Reinmar Hager, I'm the organizer of this year's meeting, and as much as I would like to welcome you here to sunny Manchester, it is actually sunny here, beautiful weather outside. We felt that traveling might be still a little bit too difficult in September for many of you, and that turned out to be true. So this meeting is going to be entirely online. It does have some advantages in that you save on fuel and costs and do something good for the planet. But it also means that more people than ever can register. And I've got two screens open here. I've got the screen where I've got my Zoom window open here, and I've got the registration coming in at the same time here. We're well over 260 registrants at the moment, and I'm particularly delighted to see so many students registering and early career researchers. We actually have also prizes for the 3 best student presentations, which we'll announce at the end of the meeting. Now, normally when you organize a meeting, as those of you who've done this at least once in your life as an academic, you have a professional conference team behind you who do all the logistics. And I asked my conference team to help me, and they said, nah, you want to do it all online, you do it by yourself. So literally I'm here in my office and I'm running the show all by myself. So my apologies for any errors in the program and in the running. It's all down to me, but I did get some help from a number of people who are here today, and I'd like to thank in particular Rob Williams, Abram Palmer, Laura Saber, David Ashbrook, and in particular Arthur Centina, who updated the CTC website constantly with all the information that I sent. So thank you very much, all of you.
And Laura Saber too. Laura did a lot too.
Yeah, Laura, that's right. Laura was very helpful as well, and she will, of course, introduce the next meeting, which will be in person next year at the end. I'd also like to thank our primary sponsors, Illumina, who've been continually supporting us for a number of years. Thank you very much indeed for your very generous support. Okay, now we've got an exciting program coming up. I'm going to hand over to Rob Williams, who probably doesn't need any introduction, from UTHSC. Our very first speaker. Again, Rob, I'm really delighted that you're able to join us after the scare of IDA going through Memphis. So it's all over to you. Thank you very much, Rob.
Thank you. Yeah, the only thing I will add is very hearty thanks to you and the other team members for assembling this, you in particular. It's definitely been a heavy lift, a little bit nerve-wracking, whether we have it in Manchester or not. We were on, I think, what they call proverbial tenterhooks. I don't even know if that's a word anymore that anybody uses, but Abe was really key in this, and Laura was key, and it's come together. I frankly like Zoom a lot, and I can say if I'm around, I will, if you want to type in questions into the chat, I will try to kind of moderate it a little bit. Maybe Abe and Laura and others of you can also just keep track of that and interrupt the speaker if time permits, although this is going to be, these are going to be fairly tight talks. Yeah, so let me just jump right in. Reinmar selected a theme for us. I don't know if you noticed that on the website, but it is essentially models for precision medicine, which is a new way to really pivot what we do in our communities. And let me share my screen. There's the abstract booklet. You should all have that. If you don't, just look through a recent email from Reinmar and team, and you'll find the abstract book. It's alphabetical by whoever's the presenter. And this is the formal title of the presentation I'm about to give with Piotr Prinz, Shonak Sen, and Gregory Faraj, all of us at UT. Genetics, Omics, and Live Fair plus Data Reanalysis using Workbooks in GeneNetwork. And let me just jump right into it. So that's, if you do want a permanent record, the entire presentation, I'll do a Carl Broman. It won't be a small link, but there's the link to this presentation if anybody wants to pull it off. And I also put it in the chat, I believe, before I started the talk. So, so here is the challenge. We know from UK Biobank, from other many large GWAS studies, that the whole point is really to handle accurate healthcare for individual humans. And individual humans are not isogenic reproductive clones like our C57 Black 6 or many inbred strains of rats. So we know we have a big problem. And I just want to make that clear, because I think many of the human geneticists are naturally skeptical about what animal models can provide, because for the last 50 years, we have been trying to develop reduced, very simple systems based on single inbred genomes. And translation, as many of this community knows very well, you can't even translate from one genome type of mouse or rat to another. So how in the world are you going to translate to humanity, 8 billion of us, with the promise that we've provided to humanity to deliver accurate, individualized, actionable healthcare? And this is a huge problem. And so I'm just going to have some dogmas or axioms, depending on which you want to pick here. But all diseases are due to complex interactions, G by E. We really need to get the right type of data in order to make accurate predictions for n's of 1 human beings. It's extremely difficult to do this. It's extremely difficult to do it in a mouse under different environments, under different treatments. But we have to develop the platform for experimental precision medicine, and that's what this talk is about. One of the key solutions is to acquire coherent data of the type that it's been common knowledge that we need coherent data going all the way back to Framingham. Most GWAS studies try to acquire extremely coherent data for one big cohort. It's not sufficiently rich really to fit any model, but it certainly is the right attitude. So Framingham, GTEx, COGA, many others are attempts to provide large coherent data sets for humanity or human subcohorts. And we have to be able to do this for mice too. And of course, JAX and several other teams are leading the charge in the rodent world. I don't think we're actually doing well enough. One of the problems is most of our data is still evaporative. Most of our code is still evaporative. So a question all of us have to contend with is, if we came back to our data in 20 years, would we be able to actually do anything with it? Would we be able to find our data? And if we could find our data, would we be able to run it on the same system that we ran 20 years ago? That's a pretty heavy lift. Most of you will be familiar with FAIR data, findable, accessible, interoperable, and replicable. Piotr and Shanak and Gregory, and actually we're not the first to use this plus after FAIR, but FAIR plus means that not only can you get your hands on the data and reuse it, but you can also get your hands on the original workflow and reuse that as well. And the reason we need this coherent massive data is so we can get to G by E, G by treatment, G by sex, G by age, all of that. So I just think, occasionally it helps to have a term that you can grab onto, almost like a rubric that allows you to have a handy metaphor. And we've tried this before using the term, Stephen Friend, coherent data, and I'm going to call it smart quadratic data or smart multiplicative data, David Threadgill also used this term multiplicative data. It just means that any vector or matrix of data that you generate for a population, whether it's human, mouse, or rat, C. elegans, Drosophila, that data be computable ideally across multiple data sets. And that's a pretty heavy lift also. So sure, it has to be findable. Sure, it has to have great data, great metadata. But that's not enough. And that's why we need this additional tier of interoperability. And toward the end of this talk, Shaunak is going to give you a tour of a Pluto lab notebook that does FAIR plus analysis of EQTL data, going back to the very first CTC in 2003 in Memphis. I presented the first QTL by gene plot using Datadesk. And now Shaunak and team can do this in real time with a recompute of under a few seconds. So you'll see what this looks like. Let me just show you what smart FAIR plus data look like in why I was so motivated by this when I started my research career in Memphis in about 1990. I knew I wanted to study the architecture of the brain, what controls the deployment of neurons to different parts of the brain, what developmental factors and aging factors control neuron number, glial cell number, et cetera. And so we've been acquiring data for many years now. This is brain weight data for about 94 members of the BXD family of mice. And on the x-axis, it's just a standard histogram with brain weight from 400 milligrams to about 500 milligrams. The D parent, the dad, is over there on the low side. The mom is on the high side. And now I can take that vector of data and I can compute the correlations of everything else that's been accumulated for this particular family of mice. So this is, again, like Framingham, but with quantitative data, although Framingham has plenty of quantitative data. But we have arguably the largest phenome, certainly one of the largest phenomes for the BXD's family at this point, with about 7,000 semi-independent traits. And so from any vector of data like this, I can compute the matrix of correlations across any number of other traits that have been measured, whether it's glucose tolerance test data, longevity, et cetera. This is going to be obvious to many of you, but I just want to say this coherent data means that when you start a study, you're not starting from scratch. You're starting on a mountain of data. And this is just showing you the mountain of data just for the hippocampus here. So these are all hippocampus versions of expression data. There are new data on proteomics for the hippocampus from Catherine Kozrowski, Rudy Ebersol, and others. Some of these are relevant from the INEA that Howard and others will remember. But up here we have data from Groningen on different subpopulations, single cell populations of progenitor cells in the myeloid lineage. So again, this is like GTEx for mouse, essentially. And I'm going to whip through the next few slides just to keep it short and sweet. But I just want to show you what you can do with coherent data of this type. So now I can take that vector of brain weight data and ask, does it covary with work that Vivek Philip did quite a while ago at the Oak Ridge? So here's open field behavior in females as part of a cocaine test, another vector of data that's quite rich. And we can ask all questions about the covariation among traits, but because of course it's in a genetic context, we can get to the causality underlying that. So the three key resources I don't know if you consider systems genetics a resource. I consider it more of a kind of paradigm in embracing the complexity or figuring out how to embrace the complexity. The problem that many of us are dealing with is on the right side of this slide, the holy grail of multi-system data integration. And I guarantee you, if you ask most biologists what those little orange and brown arrows represent linking the levels, they will not be able to tell you what those represent. They, in my view, at a crude level, they simply represent correlation coefficients. Ideally, they represent causal links. And that's, of course, where the very bottom tier are instrumental variables of SNPs, CNVs, and indels comes to the fore. And our goal, our general goal is high ends of animals or genotypes, or ideally replicable genome types, to the locus, to the gene, to the mechanism, to the treatment. But you can run it the other way around. You can start from the gene variant and do a PheWAS and look at the pleiotropy of a single variant. So this is the new upgrade of GeneNetwork 2.3. GeneNetwork is an antique by the standards of web services. It was started in January 1994 as Portable Dictionary of the Mouse Genome. It is currently, and actually always has been, the oldest web service in biomedicine. Obviously, OMIM beats it, but OMIM didn't start as a web service until 1996. So we've been plugging away at it. I wouldn't say it was sophisticated for the first 20 years, but it is now. And I have Amelie Beau to thank for pointing Piotr Prince at me and me at Piotr Prince, because we now have a world-class programmer who's been able to really lift the game over the last 8 years of collaboration with him. So the new version of GeneNetwork is rock solid code. Well, rock solid. Those of you who've used it will know it's not really rock solid, but it's pretty good. And I'm going to speed through these because you can look at it by yourself. But here's this brain weight vector. Here's the mapping that happens either using GEMMA or RQTL1 right now. RQTL2 will be coming. And you'll see Shaunak's new code. So he has some code he won't show today called FlexQTL that is really multivariate mapping at a new level. And he's going to show us LightQTL, which is also very cool and wicked fast. But this is GEMMA as a Manhattan plot, computing now very quickly for that vector brain weight data. And this is something that Reinmar and I did several years ago. We took all of the morphometric data for different brain regions, olfactory bulb, cortex, hippocampus, whole brain, whole body, cerebellum, LGN, striatum, and basolateral amygdala, mapped them all and ask a fundamental scientific question. Do parts of the brain operate as a giant balloon? They blow up and the volume changes coherently, which is an idea that Barbara Finley published in Science, or is it mosaic? And we definitely showed that it is highly independent in control. So I'm going to quit now with a terrific part of a poem by Edna St. Vincent Millay. Upon this gifted age, in its dark hour, falls from the sky meteoric shower of facts. They lie unquestioned, uncombined. Wisdom enough to leach us of our ill is daily spun, but there exists no loom to weave it into fabric. And this part gets me. Undefiled proceeds pure science and has her say, but still upon this world from the collective womb is spewed all day the red triumphant child. And she wrote that in 1939, and you can imagine what she was thinking of. But with the opioid epidemic, with all the problems we have in our world, the red triumphant child is definitely stomping around on the world, and we can do something about this. So Shaunak, I'm now basically going to stop my share, and if you want to take over, I think.
Thank you, Rob.
Good.
And may I ask how much time I have, just for clarity?
I can't hear you, Reinmar, but I think you can go probably 5. Is that reasonable? Little.
I have to unmute myself and to find myself again here. Yeah, we've got 5-6 minutes.
Okay, great.
Thanks.
Thank you all. So today is actually 901 day. 901 being the area code of Memphis. So good day to give this presentation. So I'll give you a very short window into what we have been doing with Pluto, which is a package in Julia for interactive visualizations. It's for interactive notebooks. So what was our motivation? What you're seeing here is also a Pluto notebook, by the way. Okay. The motivation is that, when we have a web service, the designers are doing all the heavy lifting. They have to guess what users want. And even if they know what the user wants, they have to make it. And it's a lot of work. And the motivation behind using Pluto Notebook is to kind of democratize it a little bit. So kind of the idea is something like the App Store. The phones are much more useful because we have the app ecosystem. So to encourage the development of user-driven apps that make the underlying data and computational engine that the web service provides more useful. So users drive what they want, and it also has the side effect of enabling the sharing of code and ideas, which is necessary for progress, promotes reproducibility, because two people, you can tweak something if you don't like the way somebody did it in a live notebook. And finally, I think it also gives us a way to write new kinds of papers instead of having a static document, which is what we have. This makes it easy to have live papers that adjust to new data, and you're not writing a new paper every 3 weeks if data is coming. Of course, there are many other ways of achieving these goals. Our idea has been to use Pluto because it was easy for us. So briefly, Pluto is a Julia package for interactive notebooks, kind of like Jupyter notebooks, if you have seen them, except that, there are some different features I'll show you later. And if you've not heard of Julia, Julia is a programming language that's very suitable for high performance computing. It's very good for developing algorithms. We can talk more about that later. The main feature of Pluto that I think is important in this context is that it is reactive, like a spreadsheet. If you change one function or variable, your whole page changes. In something like Jupyter, each cell is independent, and you can get strange things if you execute the cells out of order, or you change the order, in some way. It is simple and very pleasing, and I leave that to you to decide. And it is simple from for a programmer, not a programmer, like a programmer light like myself. I can make something very easily and show it to you. So what's our implementation? I'll show you a few examples. Piotr has created a GUIX container in the backend that fires up the environment for creating the notebook. It initializes a web server. Piotr is not, and I are not really working together other than he set it up and that's it. Everything else is done by myself and Gregory Faraj. And I'll show you a few examples of how it works in practice. So the first, this is one app. These are all running on pluto.genetwork.org. Don't go there because we'll be writing on the same notebook, but it's all there. So
So
here,
can you make your screen just a little bigger? Just make it, amp it up about 50%, the font size. Perfect.
Okay. So this is a short note on sample size, something mundane that all of you know. And this is a visualization on how sample, like the one sample test depends on sample size, false positive rate, and the effect size. So if I change the effect size, you can see how the alternative distribution in red is changing and how the power is increasing. If I amp up the sample size, the power increases because the distribution's narrow. If you increase the false positive rate you wish to tolerate, power increases. So you can see that it's very easy to do these things. And this is the line of code that is required to make a slider, just a single line. And this is a one, like 3-liner to make the power function. Here is
Shonak, sorry, could you try and wrap up, please? We're running out of time because we've got the next speaker coming up in a minute, if that's okay.
Okay, so I'll show you the one thing that I think you might like. This is analysis of the BXD population, of the spleen transcriptome using the BXD population. It's so this is real data from GeneNetwork, and what I'm going to show you is the EQTL plot, which has a plot of the positions of the QTLs. So I can change things like I can make the threshold for display a little higher, like a little bit. Well, it's going to take a second. Okay. And I can also change if I want to change which individuals to keep, I can do that and it will immediately update. It'll take a couple of seconds to do this.
And this is not from memory, it is recomputing from scratch.
Yes, yeah, that's right. It is not from memory, you are recomputing from scratch. So this is, that's it actually. So
well, thank you very much. I mean, it looks like we probably need a workshop on this or something similar to really, show the potential of that. And unfortunately, we're running out of time. If maybe anyone's got one quick question, we can try and answer that quickly. But I think maybe a workshop or something similar, sure, that would be really great. So I don't see any hands up here. So thanks again, Rob and Shonak for this. So can I move on, please, then to our next speaker who's been patiently waiting, Christoper Baker from the Jackson Laboratories. Chris, are you there?
Yeah, I'm here. Hopefully you can hear me. Awesome. Okay. Great. Well, I really appreciate the committee selecting my abstract to give a talk. I've actually never spoken at the Complex Trait Consortium, so excited to be here. I think I attended one when it was joint with the IMGC in Washington, D.C. a number of years ago when I was a postdoc in Ken Pagan's lab. Anyway, today I'm going to talk to you about work that I've been doing using the BXD resource, and it's almost entirely work from a graduate student in my lab, Candace Byers, who actually just defended 2 weeks ago. All right, so the idea here is that a decade and a half or more of GWAS studies have shown us that this is just a publication talking about the proportion of variants that's been mapped that are actually change the amino acid in genes for a number of phenotypes, and it's just clear that there's it's the lower proportion. There's fewer variants that impact gene structure, and most of the variants actually fall within non-coding regions that are regulatory in nature. So sort of the question is, if we have a single genome, we understand in populations that we have mutation, and then that genome within an individual can undergo modification, which diversifies the genome into the epigenome, which allows specialization function. And sort of my question is, how does this variation impact the epigenome and presumably potentially development? So in order to model this, I'm using mouse embryonic stem cells as a system. A lot of the work due to Dr. Laura Reinhold at the Jackson Laboratory has developed really great methods for isolation of mouse embryonic stem cells from diverse mouse strains. And so, you know, the mouse stem cell represents the inner cell mass that we can isolate and then culture as a self-renewing population. That's naive, represents naive pluripotency. And the sort of classic morphology is represented by stem cells we've derived from black 6 mice that have these nice bright edges and dome colonies. And when we derive stem cells from DBA, 2J, or D2 mouse, you know, they're much more flattened, spread out, and they look more differentiated, although functionally these are naive pluripotent because we can put them back into blast and they're germline competent. So we have these established cell lines that we can differentiate in vitro into what we call epi-LCs for epiblast-like cells. And these represent peri-implantation epiblast, where this has been considered more formative state that allows the cells to receive cues that allow them to differentiate into the primary germ layers. We can then also take these epiblast-like cells, and we have developed a reproducible method for spontaneous differentiation to form what we call these organoids called embryoid bodies, which represents sort of organization similar to you would find in the sort of near gastrulation, which have formed the 3 primary germ layers. And so today I'm going to talk about the process of cell state transition and how genetic background influences that. And so what we've done is we've collected and grown replicate, independently derived replicates for B6 and D2. We collected RNA-seq data to look at transcript programs and attack-seq data to look at regulatory element activation in both ESCs and epi-LCs. And then we've differentiated these to EBs and collected single-cell RNA sequencing data to both identify the cell types that are present in these embryoid bodies, as well as look at the proportion of them. So just starting with RNA-seq data, we find, so each dot represents an independently derived embryonic stem cell from the line. Blue is going to be B6 and orange is going to be D2. And so we find that the major component of variability in this data set is, of course, cell state transition between ES cells and epi-LCs, but there's also large contributions to genetic background across a number of extra principal components downstream. And when we want to ask, like, sort of what's driving these differences within a state between genetic backgrounds, we look at the pluripotency factor expression of naive transcription factors that maintain pluripotency. They're not really different between the strains. But when we look at the attack-seq data and look at the accessibility of their motifs in the genome, we really find that a lot of these transcription factors like NR5A2, ESRB, OCT4, or POU5F1 actually have, they're much more accessible. The motifs are more accessible in the B6 background compared to D2, which is consistent with the morphology. So to dig down into the data a little bit more, we developed a model where we created a term for both state, strain, and an interaction term between state and strain to look for genotype by state interactions in gene expression. And so we find a lot of interesting types of changes in gene expression. Here's just 2 examples for this NR5A2, which is a transcription factor involved in pluripotency, where it's high and comparatively high in ES cells, but B6 is turning off that gene much faster than D2 when exiting pluripotency, where another example for SOX1, which is a marker for neuroectoderm differentiation, we see B6 turning this gene on quite a bit faster than D2 at the same time frame. So these are just examples, but we wanted to look at sort of modules. So we used a method to identify modules of pathways as they shift between. We took all of our genes that had significant genotype by state and developed modules for these. And we again find, I'm just going to highlight 2 modules here. Module 4 contains a bunch of genes involved in retainment of pluripotency, and module 5 is involved in neurogenesis. And sort of the eigengene of these modules behave similar to the individual genes, as you would expect. Again, so what we sort of generally see here is that B6 seems to be ability to leave pluripotency faster while also turning on genes associated with differentiation. Sort of confirm this, I mentioned we grew these embryoid bodies, which are organoids, and for replicates in both B6 and D2, and perform single cell sequencing where you got these nice clusters. I like to call this, anybody who has kids or did it as a kid, you make a turkey at Thanksgiving with your palm, you know, but it's more like a dog head. Anyway, it's our dog turkey plot. And so when we try to understand what these different clusters are, we can look at marker genes, identify vascular endothelium, primitive erythrocytes, mesendoderm, definitive endoderm, all these are examples of primary germ layers. But I want to draw attention to this cluster 11 here, which represents neuroectoderm, which is, when we look at the cells that are in here and the genotype of the cells that are in this cluster, we see that's by far more made up of B6 cells, again, which helps to support the idea that B6 cells are transitioning to a neuroectoderm fate at a higher portion than D2 in this system. And there's sort of this classic idea in ES cell culture and field that neuroectoderm is the default cell fate, but of course everybody who studied that and sort of made that default claim has been studying cells derived from B6 or 129. We have been able to confirm this using repeat organoid cultures and using FACS looking at protein markers instead of gene expression, and we get the same effect. So being geneticists, we're interested in what's driving this. So we took the BXD panel and then derived, these are supposed to be like, you know, cell culture plates, but we derived embryonic stem cells from individual BXD mice. And then again, collected both RNA expression shown here, but also attack-seq data from individual BXDs. And again, the primary determinant of variation is going to be the cell state, but you can also see contributions to genetic background, and the BXDs are sort of ranging in between the parents, although as they begin to differentiate, they, you know, some of them are, you know, have wider variation. So we did QTL mapping, and of course, each dot represents either a gene, or in the case of chromatin, each dot represents a chromatin accessibility peak that we identified in the genome. And of course, we have lots of cis variation mapping, but we also find a number of these prominent trans hotspots that are suggesting coordinated control of both chromatin organization as well as gene expression. I'm just going to highlight 2. So we did this in both, the data I'm showing on the right is ES cells, but we also did it in epi-LCs. And what I'm plotting here is just the number of targets based on the QTL. It's not a log map, but you can see that a lot of the same loci appear in both ESCs and epi-LCs. But I want to draw attention to this chromosome 12 region, which in ESCs have a lot of chromatin QTL targets, very few chromatin targets in epi-LCs, where in epi-LCs we have a lot higher change in gene expression that map back to the same locus. And in fact, a lot of these chromatin targets that are variable and controlled by chromosome 12 in ESCs are in proximity to the genes that change in epi-LCs. So when we ask what those genes are and we begin to explore if they have any common pathways, we find that about half of the genes are found in our neurogenesis module I talked to you about earlier. That's in the genotype by state interaction. And they're all driven, when the QTL is B6 at 12, all these genes are expressed higher. Again, suggesting that perhaps this chromosome 12 region is influencing the B6's ability to more rapidly adopt neuroectoderm fate. Okay, so in terms of what are some candidates at these regions, we don't really know what the candidate is 12 yet. We're still interested in that, following it up. But I have some data for a different one. So this on chromosome 4. You know, one of the questions we asked is what kind of transcription or DNA binding factors are bound at these variable chromatin regions. And so we downloaded a data set from this Systrome database, which seeks to curate every single ChIP-seq that's ever been done, both in mouse and human. So we downloaded their data set, filtered for those that were done in ES cells, and looked at what was enriched there. And so we found the highest one is this TREM28, which is a repressor, which I'll talk further about. We also found enrichment for known transcription factors important for pluripotency. And so we redid our own ChIPs in these as well, using our own cell lines. And what we find generally is that, for example, like B6 has higher enrichment of TREM28 at these binding sites compared to D2. And then conversely, the other transcription factors are less enriched. And I'm going to show you an example of what that looks like specifically here. So there's this locus on chromosome 18 that is controlled by this chromosome 4 QTL. Both two genes, RYOC3 and RMC1, have high LOD scores that are that map to chromosome 12. And then all these black boxes here are CA QTL, so variable chromatin accessibility regions that are in the same locus controlled by the same QTL. The one with the highest LOD score in the middle here has really high binding of this TREM28 factor in B6, and subsequently low accessibility in D2. And that's again shown here, where the B6 parent is not accessible and it's open in D2. And when we look in the F1, we find that both for gene expression as well as chromatin accessibility, that we see this sort of dominant repressed phenotype.
Chris, sorry, could you try and wrap up, please?
Yep. Okay. So I'm just going to say that this region is covered by crab-zinc fingers, which bind TREM28, determine where they bind. And another group led by Todd McFarlane has created a knockout of this region. And essentially, whenever we look at their data, we see reactivation of our QTL targets. So you knock out these crab-zinc fingers in B6 ES cells, we get reactivation of these targets. And I'm just going to end there. So we believe that at least this QTL is controlled by these crab-zinc finger proteins, recruiting TREM28 and creating heterochromatin. And I'll skip that. And I just really want to acknowledge Candace Byers, who's been a great graduate student, and we'll be sad to see her leave, and the rest of the lab and collaborators at Jax, Laura, Steve, and Dan. Happy to take questions if I have time left.
Yeah, thanks. Time for one quick question. What we got here?
So the chromosome 12 transband, it looked like it's pretty close to AHR, which is a big dude transcription factor, obviously. It's known to be highly polymorphic between B and D. As a matter of fact, the black 6 has a weird mutation in AHR. So there you have a transcription factor. I don't know if it operates that early in development.
Yeah, so thanks for the tip on that. I haven't actually looked about that. AHR, this is really proximal end of chromosome 12. It's a recombination cold spot, unfortunately. And so it's a fairly large block, and it's a kind of a poor annotated region of the genome. It's highly variable across lots of mouse strains, including D2 and B6. So I haven't looked at that specific candidate though, but I'll follow up on it.
Very, very cool talk. There are very few hardcore developmental biologists that have the bandwidth to add true genetics to their studies. So just a big bravo from me.
Thanks. Right. I'm not sure if I'm a hardcore developmental biologist, but I'm trying.
That's great. Thanks, Chris. Okay.
Yeah, thank you, everybody.
Thank you very much for that. Let's move on to our next speaker, Abe Palmer, please. Yes.
Yes, that's the view you expect. Okay, great. So I really want to thank the organizers of this meeting. It's a terrific effort. Reinmar is obviously the key person in doing that. And I think it is great that we're meeting online, and I think everybody should think about the environmental benefits of trying to transition our culture to have more online meetings and to value online meetings, because I really think the previous model of flying all over the place is unsustainable. I'm going to talk generally about this big project that I've been involved with for the last many years, using HS rats, outbred heterogeneous stock rats, for GWAS studies, particularly with the focus on drug abuse, but as you'll hear, actually using them to study a variety of other traits. Over the next few days, 3 days, you're going to hear at least 10 talks I counted in the program that talk about these HS rats. And so one of my goals is to sort of put those in context and to give a little bit of a background about the overall use of HS rats and our center's activities to support that. A website that you can visit, and I'll get back to that website later. So I think that what we're doing in HS rats has become a little bit like the US All of Us study or the UK Biobank study, where we have a tremendous amount of data on individuals, phenotypic data on individuals for whom we also have genotypic data. And it's the goal is really to create an ecosystem, kind of like what Rob was describing for systems genetics, an ecosystem where different data types that are generated by different investigators can all be jointly analyzed and integrated to give a greater understanding than you would have with a number of freestanding studies. And fundamental to this approach, as I'll describe in some of the next slides, is a breeding core run by Leah Solberg Woods that distributes HS rats. So these are outbred rats, as I'll describe in the next slide. Distributes them to a variety of labs, mostly in North America, but we are shipping them currently to Italy also, and certainly have the capacity to ship them wherever they need to go. And then paired with a genotyping and analysis core that lives here in San Diego that I'll also describe, that makes it possible to genotype them on a uniform platform and then to integrate the data critically into a database that allows for a lot of interesting analyses. There are a few other projects here, which I'll briefly allude to, but which are looking at structural variants in single-cell RNA-seq that are sort of supporting a lot of these, but that break from the general model of shipping rats to phenotyping centers and then sending that phenotypic and genotypic data to an integrating center. So these two cores are really critical to that, and I'm going to describe first Leah's core. And I should say that Leah was maintaining these HS rat population in North America, the only North American population, before the center was ever formed. And the fact that she had that colony was critical to our ability to put this together. But even before Leah's time, back in 1984, before I think some of the people on this call were born, present company accepted though, 8 inbred rat strains were mixed specifically in order to make a genetically diverse rat population. At the time, they wanted a genetically diverse rat population for selection studies, but I think they also recognized that in general, inbred strains weren't the only way to study genetics. And that has been maintained, I think, now for 92 generations, but we're approaching the 100th generation of that outbred population, and it's passed through a number of hands, as I describe on this slide. So it was initiated in 1984, and then Eva Rady had it starting in 2003. And then since I think 2006, Leah has been running this colony, originally when she was at Medical College of Wisconsin, but now she's moved to Wake Forest. And it was transferred over there in 2016. And then actually at the beginning of the pandemic, we decided it was critical to freeze down embryos of the current generation. And so Leah in particular was able to get 75 breeding pairs and have the embryos frozen down so that if some tragedy should ever befall this colony, we'd actually be able to reinitiate it from those embryos. And that was particularly important. For some of this time, there was a parallel colony that lived in Barcelona, but that colony no longer exists. And so Leah is now the only person in the world maintaining this colony, which made it really particularly critical to freeze it down. The other critical part of this center is, which lives here in San Diego, is our ability to genotype and analyze phenotypic and genotypic data that come from HS rats. we don't rely on a microarray. So people who are familiar with the mouse world and outbred mice in the mouse world, particularly diversity outcross mice, rely typically on a genotyping microarray to get their genotypes. We have never developed such an array in rats. And I would argue that that's a good thing because it gives us a lot more flexibility to use our approach in different rat populations or even different species. But we actually used a genotyping-based or sequencing-based technology to genotype the animals. And we're able to sequence at very low coverage, about 0.25x, and still are able to get good genotypes with about a 1% error rate relative to deeply sequenced or other kinds of gold standard genotypes. And that also allows us to accumulate increasingly deep coverage across all of the haplotypes that live in this population, because whereas we're only getting 0.25x per individual, we're doing this in thousands of individuals. And the nice thing about this approach to genotyping, we originally actually developed something like this in mice and then only subsequently applied it to rats. But because it's sequencing based, it's very easy to pivot and go to either a different rat population. So for instance, we do this in Sprague Dawley rats, another outbred population, but also in other species. And you're going to hear 2 talks later in the meeting applying this approach to zebrafish and a kind of a funny critter called Paramiscus californicus. And so this sequencing-based approach is really one of the key things that make this center possible. And all of the different phenotyping centers are genotyping on that same platform, which is important for integration. The other thing that we do is we store all of the phenotype data in a fairly sophisticated relational database. And here's the database team as it stands now, led by Apurva, who will be talking later, though not specifically about the database. But we bring in all of the pedigree information about these animals, all of the shipping information about the animals, the phenotype information, and critically for most of the behavioral phenotyping, there are a lot of raw files that are typically processed into individual phenotypes, or maybe a few phenotypes that we think are important about those animals. But we bring in the raw files and then write R scripts on top of those within the database to extract what we currently think is the right phenotype. And that allows tremendous flexibility because when we need to, we can change what we're extracting from those raw files without having to ask the individual investigators to reanalyze everything. And so that makes the database a lot more flexible and I think powerful. We also have, of course, all of the genotype data there, and we have other omic data types that we're collecting about these animals. So for instance, microbiome, and you'll hear about from Amelie, some of her work with microbiome. Obviously, a lot of gene expression work, as I'll describe in some of the subsequent slides. And then other kinds of omic data that we have, and metabolomic, et cetera. And all of that is stored within this database. And we actually just yesterday got a supplement, which I think maybe Rob has applied for a similar supplement from NIH to try to make this data more fair, more findable and usable for other people, particularly for people using machine learning based approaches. So this database lives on the cloud. And we also then have an automated pipeline for the analysis of the data. And so one of the key things that we've put together is a pipeline that allows us to say, look, here's the phenotypes, here's the genotypes, go. And then we get from that the GWAS, but we also get the fine mapping locus zoom plots. We also get reports that include the EQTLs that co-localize with the hits, the coding variants that co-localize with the hits, genetic correlations that allow us to correlate traits that are measured in separate cohorts of animals, PheWAS, that is other things that seem to live at that same locus, other traits that we've also mapped to that same locus, et cetera. And that's all done in an automated way. It's an HTML document, which means that it's somewhat interactive. I'm inspired to hear Sonak's talk and hope that we can make it increasingly interactive with time. But it's already a somewhat interactive report that gives people essentially all of the analyses that they would need to put together a paper. And so Rob mentioned a mountain of data, and I just wanted to put a picture of a mountain here to say that we are generating another mountain of data. The project itself, I won't say much about phenotypes because there are a lot of them. The P50 grant itself has 3 phenotyping centers, and we'll actually be hearing from both Paul and Howe's lab about results that they have from those. But what's really been critical is we've been able to use these resources, the ability to ship rats to people, the ability to genotype and analyze rats, to support a variety of other grants, most of which are written by other people. Many of those you'll see are NIDA grants, but many of those are not NIDA grants, also are grants using other funding institutes, other kinds of phenotypes. And that's really what's giving us that richness. And in that earlier slide that I showed you, where I said this is becoming a little bit like All of Us or UK Biobank, it's the addition of all of these other grants that are also using these core resources that's really building the mountain, the hill of data that we had into a mountain. We've also made a practice of cutting up these rats when we're done with them. So our studies focus on behavior, and that means that at the end of the study, we have a lot of tissues that we don't really need, but that we can share with other people. And so we've been sending out lots of different parts of rats to labs that are interested in studying parts of rats. And there may be people on this call that would like to start receiving parts of rats in the future. Some of those have now turned into funded grants on their own right, but many of those are unfunded. And because we're able to give them the tissue and then analyze the data essentially for free, in some cases, it's not necessary for them to write grants in order to do that work. We really emphasize the integration of gene expression, and you heard already in some of the previous talks of kind of the importance of gene expression as the likely cause of most of the complex trait variability that's mapped in these sorts of studies. And so we think about the relationship between SNPs and phenotypes as the primary thing that we map with GWAS studies, but we imagine many of these are actually mediated by the effect of the SNP on transcripts, and it's the transcript variation that changes the phenotypes. In the very next talk, you'll hear from Dan Munro about some of the latest work that we're doing on EQTL mapping. And then from Haki Im, you'll hear on Friday about some more sophisticated things that we're doing with some of that EQTL data to actually come up with polygenic transcriptomic risk scores. And I won't say more about that, except to say that it's coming. We have access to an enormous number of tissues. So Rob and his BXD database have, I think, still many more gene expression data sets, but these are all RNA-seq data sets, some of them with pretty substantial sample sizes that we have access to and that we're trying to organize again in a fair kind of a manner so that other people can use this. And as Dan will describe, we're also putting together a kind of a GUI-based interface that'll allow people to look at this data for the particular genes they're interested in. Just to briefly kind of show how we're putting all of this together, here's a recent publication led by Apurva, where we looked at body weight, which was something that we were measuring in a number of different centers. And so we had a really very large sample of body weight data. This is a porcupine plot showing several Manhattan plots superimposed on one another, just trying to make the point that we find a lot of stuff. Some of these hits are absurdly significant because of the sample size. And we also see a lot of pleiotropy where different traits are mapping to the same locus. We can then do locus zoom plots of those, and this is all just pulled from the reports that we generate. And in these locus zoom plots, we see the relatively fine-grained resolution that we get, and we can see the relationship between the associations and the genes within those regions. And then significantly, what we saw when we looked at this data is that the number of findings that we have is not linearly related to sample size, it's actually exponentially related. So as we grew from the sort of conventional sample sizes for these sorts of studies, maybe 500 or 1,000 rats, to these much larger sample sizes, we saw that the increase in the number of findings was not a linear function of sample size, but grew exponentially. And I think really emphasizing the need for people planning these kinds of studies to not just think about the power you need to make one finding, but the power that you need to really get the full value of the study. And those sample sizes, I think, are going to be larger. And I think this would probably apply equally to mice, although we've done it so far only in rats. So there'll be a number of talks. I grouped them into two basic categories: talks about phenotypes, and I won't say everybody's names here, but you can see a number of talks over the next 3 days, and then talks emphasizing the need to integrate the rat data that we're generating with other species, particularly human, although I think there's an equally good case to be made for integrating with mouse. The talk from Leah, I should say, is actually independent of the center, but uses HS rats, but it's her data, her analysis. And then we're also doing some network work, and again, Hakeem's talk. I want to acknowledge the growing resources available with inbred rat strains. I won't be talking about those, but there are a number of talks about that, and I think that's critical strength for the rat community. And then I just want to thank all of the people that are contributing to this work and invite people not yet involved to reach out and join us if you'd like to get involved. And that's all I had to say.
Lovely, great. Thanks very much, Abe. We've run a little bit out of time for questions. Maybe people can put them in the chat and you can look at those if that's okay. So let's move on to our next speaker, Daniel Munro. Daniel, are you there? Yes, you are. I can see you. How are you?
Yes.
Very good. Right, over to you.
Hello, thank you. Okay, so yeah, hi everyone. I'm Daniel. So I'm a postdoc in Abe's lab and also in Pejman Mohammadi's lab at Scripps Research. And so I'm going to show you our work on mapping and analyzing EQTLs in multiple brain regions of heterogeneous stock rats. And so as you all know, as Abe was talking about, rats are an important organism for mapping the genetic associations with different behavioral and physiological traits. But often with these GWAS loci, we don't know what the biological basis of them is, if there are any causal genes involved. And so the big motivation for this study is to map the genotype expression associations, called expression QTLs, and across the rat genome. And so this will allow for different things like identifying regulatory elements and being able to co-localize these with the GWAS QTLs, and that can identify putative causal genes. And in addition to helping understand the biology of it, involving genes can increase the relevance of these results to human biology. And so to do this, we obtained RNA sequencing data from 5 different regions of the rat brain from 88 heterogeneous stock rats. And so Abe had also talked about what the HS rat population is like, but this mosaic of different founder haplotypes provides us with a lot of genetic variation, which we obtained the genotypes from genotype by sequencing, and we quantified the transcriptomes from the samples from these 5 brain regions. And then this is showing what the haplotype level diversity looks like. So using the genotypes, we estimated the portions of each animal's genome coming from the founder strains. And we were kind of surprised to see that this was less uniform than we expected. But after we did some simulations, we found that this really falls in line with what we would expect from the amount of genetic drift you would get over time. But overall, looking at the genotype level, this leaves us with a lot of genetic diversity. If you look at the minor allele frequencies, they're fairly uniform and overall a lot higher than you would get with some populations like humans, and that gives us a lot of statistical power. So using this data, we mapped expression QTLs. So we modeled this loosely off of the GTEx pipeline. So this is the genotype tissue expression project that was conducted over dozens of human tissues over the last decade or so. And so we used single SNP linear regression using TensorQTL, and we mapped associations between every variant and genes expression levels. And so you can see that the vast majority of the strong associations occur with variants close to the gene. And these are, we call these cis EQTLs because they're putatively involving the cis regulatory associations. And so we focused our analysis on these cis EQTLs that are within a megabase of the gene's transcription start site. So overall, over these 5 brain regions, we identified cis EQTLs in almost 8,000 genes. And using stepwise regression, we were able to identify multiple independent EQTLs for some of these. But this just gives you an idea of how much those EQTLs overlap across tissues and how many of them are tissue specific. And then the distance from the eSNPs to the transcription start site, you can see they're concentrated closer to the TSS within this cis window, which is consistent with just where enhancers and promoters tend to be located. And we validated these EQTLs using the orthogonal model of measuring the association. So instead of just the number of alternative alleles and the total expression, we can also look at the heterozygous individuals and measure the allele specific expression for that SNP. And using these 2 different measures of effect size for the EQTLs, we confirmed in each brain region that the effect size is correlated pretty well, as we would expect. We also wanted to test whether it would be necessary to use linear mixed models in this context of cis EQTLs in HS rats. And so this is a way of adding a random effect term that models the genetic relatedness between the individuals. But overall, we found there's actually very little effect on the p-values of the cis EQTLs when we added that term. And another way of showing this is that whatever p-value cutoff you have, you get almost the same results, the same set of EQTLs. And so we think this has a lot to do with the fact that there are some closely related individuals within this cohort of 88 rats, but overall we don't see major clustering in population structure, which is a big motivation for linear mixed models. So for this reason, we just went with the GTEx approach of using the fixed effects linear model for our main results. And so finally, because we expect these EQTLs to be important, be useful for the rat genetics and genomics communities, we want to make it available. So we've initiated this Rat GTEx portal, which is available at ratgtex.org to host the data. So this is inspired by and with code adapted from the GTEx portal, which is for human tissues. But we are using a uniform data processing pipeline. And so that when we, on an ongoing basis, we'll add data sets from different tissues, from additional studies, but we'll be able to compare results across the tissues and studies. So this portal provides some interactive data visualizations, such as the one shown here, and as well as data downloads and an API for the data, which, as Rob was talking about, will follow the FAIR principles using the open AI framework. And here's another visualization that it shows. So there's a screenshot of one showing all of the EQTL results for a gene. And so you can see it shows all of the tissues, has different ways to filter. You can search by gene. So I think it has a lot of cool features, if I may say so. So to conclude, I've shown how we have mapped and analyzed EQTLs in 5 brain regions in heterogeneous stock rats. And that EQTL discovery, both the methodology and the results, resemble what has previously been shown in humans, but with some important distinctions, such as the relative importance of population structure versus the importance of LD structure in the genome. And we have built the Rat GTEx data portal and have these 5 tissues added to it to provide these and additional projects to the rat genetics and genomics research communities. So I would like to thank the other members of the Palmer Lab and the PEDGE Labs for their support and ideas, as well as to the rat genomics and genetics communities, which we're glad to be a part of. And then this work was funded and organized by the NIDA Center for GWAS in Outbred Rats. So thank you, everyone, and I can take any questions now.
That's great, Daniel, super. Thanks very much for this. You're the last speaker in our first session, so we've probably got more time for questions. There's no speaker now until past. So, you know, you can step out, take a break, but I think we can also use that time for questions. I do have one very general one, possibly to both you, Abe and Daniel. It's about power in that system. So what kind of sample sizes do you need for mid-effect QTL to detect, and what kind of sort of biological replicates do you have to use?
Yeah, we did find with 80, I guess for each tissue we had around 80, and we compared the number of EQTLs we found to how many were found in like GTEx tissues, for example, and we found that there's definitely this trend, as Abe had pointed to with GWAS studies, that our sample size compared to GTEx was on the low side, but that as you increase that, you can get many more. Basically, the more samples, you can probably find EQTLs in like any gene that there's variation near. And so, yeah, I think
so
as
Dan,
we go on.
In case somebody wanted a specific number, no, I think your answer is great. I think if somebody wanted a number, one of our low numbers was about 75 or 80, I think, for one of those tissues. And I think it's right. You still are finding thousands of EQTLs, even with that small sample. But I think, don't you and I both think that even larger samples would be better? Is that a fair synopsis?
Yeah, it should scale up at least linearly. I think we had another project with around 53 samples, and there were a lot fewer EQTLs. So I think if you're to pick kind of reasonable minimum, it would be somewhere between 50 and 80 samples. But
yeah, and I think we've been proposing larger samples in the low hundreds, kind of, and have a grant in actually with Hao Chen, led by Hao, where we'd like to get to much larger, like more than 1,000 samples, because I think that would really unlock a lot, even though we wouldn't be able to do that in every tissue. I think it would unlock a lot of potential to go to really dramatically larger samples. So far, I think the biggest sample we have is about 350. And Dan didn't present that. We're just kind of analyzing that now. But
yeah. Yeah, I think there's a trade-off to be made between like how many different, if you want different brain regions versus more samples for a smaller number of regions to get higher power. So that's something important to think about.
Okay, that's great. Thanks very much. I think you can just, continue the discussion without me moderating. I think that's fine. We've got a few more minutes. In fact, 14 more minutes. Rob, you're on mute.
Yeah, Dan, really lovely presentation. I love the Rat GTEx, excellent idea.
Thank you.
One thing that would be fun, and this is, Abe and I have been pinging back and forth on this idea, to what extent live computation is practical, and when do you have to database the actual vectors of results. And we generally database the vectors of results because it's just too slow to do it in real time. But what Shaunak showed just a few minutes ago, the light QTL really can do this in real time, but I'm not sure to what extent it can do it for, say, a minimum of 100,000 markers, which is probably what you require. What is the lowest you think you could skate by on in terms of marker number to get a reasonably good eQTL map, given the LD block size?
I think number of markers. Yeah,
I mean, go ahead, Dan. Sorry.
I haven't thought. Yeah, I haven't thought much about what the minimum could be. I've tried it like when I was mapping trans eQTLs with Gemma to test that. I was using like pruned genotypes.
Yeah, if you just threw everything with an R squared of greater than 0.8 away and just kind of kept, I won't call them tagged SNPs because they're not tagged anymore. I'm not sure what the heck you call them. But anyway, keep it at 0.8, 0.9 between adjacent SNPs, see what you can get down to, see whether you can replicate your full imputed genotype set. And then it might be fun to see whether you can actually get kind of live eQTL plots, because I would love to be able to fit crazy models. I would love to be able to fit the cis eQTLs and get rid of them.
I want to get to Amelie's question, but Rob, I think the key there would be just to focus on the cis window. So since most of the action is in the cis window, that's a really small number of SNPs and would be really feasible to do the mapping there. For the trans mapping, there'd have to be some trade-off like what you're describing, sort of a coarse map of SNPs to make it feasible. But Amelie has been waiting patiently for a while to ask a question.
Yeah, I think there's an interesting discussion to have about the reproducibility of inbred versus and outbred resources. So we heard Rob talk about BXDs, which are inbred, and Abe talking about the P50 HS rats, which are outbred. And Abe, you showed 15,000 HS rats being used. Of course, not all the phenotypes are collected on all of them. So can you say a word, I'm thinking about your AIL paper where you looked at different generations of the same population. Do you find the same genetic effects in that case? Do you find the same genetic effects in two current HS populations?
Yeah, we do have some data, and actually I think Apurva is going to talk later about locomotor behavior, which has been measured in different ways, sort of in different populations. And so, as you would expect, they're correlated, genetically correlated, but not identical. And that could be due to measurement differences. We have some other data sets where we made more of an effort to kind of standardize things so that it was more comparable. There are always going to be effects of center, and thinking back to the old John Crabb paper about inbred strains and the effect of phenotyping center, I think there's always going to be some of that. We haven't looked, though, and it's a good idea, at effects of generation. And I suppose it is very plausible that over generations, there are differences in the phenotype. Of course, there'd be some changes in allele frequency if you look at a span of a lot of generations, but there could also be environmental or other reasons that there would be differences or kind of effects of generation. I don't think we've done a good job of trying to study that yet.
So overall, would you say that inbred lines are more reproducible than outbreds? Would you say that is still true, or do you think it's reasonable to use outbreds over generations just as a reproducible population?
I guess I think that sometimes you want to study the animal and you want a phenotype about an animal. And of course, inbreds have a big advantage there because you can have the same animal many times, sort of, right? The same genotype. But often we're interested in the effects of alleles, right? And what we really try to study with any kind of a GWAS approach is what does this allele do to my phenotype and what, to a number of phenotypes, maybe including gene expression. And I think that outbred populations are great for studying the effects of alleles because those alleles exist and they're segregating in all of the animals that you observe. And so they're useful for studying that. So I think it depends on what the question that you're asking is to some extent. I think in many cases, we want to study alleles and not animals, not individuals.
We're trying to dig out this paper that came out last year that looks at variabilities, phenotypic variability between inbred and outbred mice, and actually shown that there's as much variability.
Yeah, I was thinking about that also.
I'm going to put it in the chat if I can dig it out. Let's see if I can find it. Got it somewhere here.